Here's what the Philippines looks like. Okay, is what we see today.
Sorry, we've started late, but we had forgot. So
no problem, no problem. But back in the Pleistocene, by this clue is that 120-meter bathymetric underwater contour back in the Pleistocene that sets up these 5, 1, 2, 3, 4, 5 large aggregate island complexes that divide and partition the diversity in the Philippines, and they're all each like separate little, completely different faunas at this point. And they have some overlapping species and some exceptions, but it's pretty striking to think that the Philippines actually did look like this and formed these big aggregate islands that have since fragmented and then refused and fragmented. And and this has formed, the basis of this dominant four-year paradigm, which has been invoked to explain the commonalities of vertebrate species distributions throughout the archipelago, but also as a mechanism that could have generated, partitioned, and maintained species diversity in these incredible land vertebrates throughout the archipelago. This has really become the central theme, and books have been written about the story. It's fueled the research program of mammalogists Larry Haney and Eric Rickert and all of their collaborators. It's picked up in the textbook that we use to teach biogeography. Is a great classic example and a really prevailing theme. And these quote, you see quotes like this that really that wrap it up as something that probably had an impact that was beyond the other sort of traditional biogeographic barriers or factors. So while I run this animation, there's a copy of a heavily cited paper that first lays out this story early on. Let me see if I can move this around so I can see. The clue here is understanding the permanent isolation of islands. So these islands that have deep water trenches around them, so they were permanently isolated from surrounding islands. That's the partition of the archipelago. And then the other is the possibility of this species pump possible mechanism of repeatedly fragmenting and fusing and fragmenting and fusing islands that do have a kind of a level of endemism throughout those islands, and species that you find on these islands and are shared among these 4 islands and nowhere else in the archipelago. But then within some of the pikes, we find pockets of diversity like northern Masbate and southern Negros and the island of Cebu that have their own endemic species as well. And that looks like those species may be the things that were produced by the species pump action of falling and raising and receding. Are we going to do this again? There we go. Whew. Okay, sea levels. There you go. Okay. Well, it turns out there's other ways to divide the Philippines as well. I just ran across this recently and thought it was pretty funny with regard to the weather system, the prevailing religions, who's laying claim to the Philippines, and the different groups, and then the difference between people who live in Manila and the rest of the country. So now I'd like to show you some groups of many groups that do obey, with some exceptions, this sort of prevailing distribution paradigm. And I'll tell you about the group that I conducted my master's work on in Sheldon Gutman's lab, which is the Cygnatic Complex. I'll go through this kind of quickly, but it's a beautiful group of frogs that are distributed throughout Southeast Asia. And in Gutman's lab, I was studying a phylogenetic analysis of 42 Allozyme loci, so very old school. And here's a figure from that paper where I was trying to infer something about how these species may have colonized the archipelago, either into the western, as you can see up here, or the eastern, the red here, different island arcs of the Philippines. And I wanted to know something about whether it was potentially 2 invasions of the archipelago or 1 invasion and 1 back dispersal. And with our non-quantitative biogeographical techniques at the time, there wasn't much we could do with it, right? So this is something I always wanted to come back to. And in 2014, with the big multi-locus Sanger data set, I was able to. And we were inspired by this paper by Jared Diamond and Gilpin that was about a biogeographic umbilici and the origins of Philippine avifauna, which allowed us to formulate the dual umbilicus hypothesis, the idea that invasions of the colonization of the archipelago might have gone in 2 different directions, and Filipinos love acronyms. So I wanted to actually publish this and get it out there so that at some point I could respond to it. So I'll skip ahead to the punchline with some massive new genomic data and interest and information from a new probe set that I'll talk about later, the work of one of our students. We've revisited this same story once again, and now we found that Pulkrana, it looks like strong support for a single invasion of the archipelago, which allows us to conclude to reject the dual umbilicus hypothesis. And what we find is that what has resulted is this geographic radiation where you find an endemic species, color-coded here to match the tree, endemic species on each of the different major landmasses throughout the archipelago that sort of perfectly match the Pleistocene or the Pike paradigm. So there's an example of a case that a phylogeny that tells us something about the evolutionary history and that results in these distributions that are so characteristic of the archipelago. Now, the one exception to that rule in this group is a species that I found in 1991 on Mount Busa on the basis of one specimen, and draw attention to the possibility of sympatric species on the same large geographically complex landmass of Mindanao. And I found that those were fixed allozyme variants on a starch gel lab in Sheldon Gutman's lab. And I hung onto that specimen for almost 25 years before I described that as a new species, even though, and it was this analysis that gave me the go-ahead, was that this species from Mindanao didn't come out nested within Grandocula, and its phylogenetic placement was a key bit of evidence that gave me the courage to go ahead and describe it on the basis of one specimen, which I don't recommend. It's pretty nerve-wracking, but this species actually just recently been rediscovered by a student in central Mindanao, and I'm delighted to say that I was right. Here's another one. This is an endemic genus of frogs, Sanguarana, that entered the Philippines from the west by this mechanism of the Palawan microcontinent block, which is something that is really interesting and has resulted in a big geographic radiation throughout the archipelago with endemic species on each of the Pleistocene aggregate island complexes. We were interested in resolving this question of, at the time of the entrance into the archipelago, did they first colonize to the south and then disperse to the north, or the other way around? And we're also interested in this possibility of a deep coalescent event in two species that appear to be nested inside the widespread Sanguarana luzonensis. And this is a former postdoc, Young Chao Su, who with some genomic data from UCEs resolved that question and showed that we were just looking at some lineage sorting in these species that were outside luzonensis and are not nested inside with it at all. And he was able to reconstruct the key nodes that allowed us to get to this answer of an initial colonization of the southern parts of the Philippines, speciation associated with sort of a progression rule dispersal up into the north of the archipelago that gave rise to an archipelago full of these frogs. And the exceptions are things like the island of Luzon, where Sanguarana luzonensis is widespread throughout the archipelago. And then you have these cool high elevation montane species that Jim will probably remember. This is Sanguarana aegarota that occurs in the Cordillera of the eastern side of the island. There's a species, Sanguarana tipanan, that Jim and I described, that occurs along the Sierra Madre on the did I say western? The western side, and now on the eastern side. And the coolest one was Irantipantata, this frog that occurs on the one mountain right between those two mountain ranges, Mount Palali in the Caraballo Mountains, and matches the color pattern of the Scooby-Doo Mystery Machine. And so Raul Diaz, a former Berkeley student, pointed this out, that Scooby-Doo fans all over the world were pretty excited when the species was discovered and described. But this thing that occurs on this U shape, there's one side in the Sierra Madre, one in the Cordillera, and one right between, looks like sort of a U species, if you will. And it looks like parts of a ring species in some ways. Okay, let me go right. So those are some examples of groups that seem to obey the pattern with some exceptions. But let me go right to these ones that just defy it wholesale, which are small species that are endemic to these tiny little land masses. For the last couple of years, I've been working in collaboration with researchers at Ateneo de Naga University in Naga City. And so when I come into Manila, we stay in Makati and have this wonderful view of the Pasig River. You can see how densely populated it was there. And the last time I came in through the country, I was traveling with Justin Bernstein, who is a PhD student in Sarah Ruan's lab, who's here tonight. He's working on Homolopsid snake evolution, and we were after the Microlepis, the Cerberus microlepis, which is this endemic to one particular lake, and maybe an example of invasion of fresh water from estuarine ancestry. And there's Justin with his first Cerberus microlepis when we finally found the snake, so pretty cool. But the news wasn't great at that time in southern portions of the archipelago, and we were happy to get out of Manila. So we flew from Manila down to right here on the edge of Mount Isurog, which is Naga City. And let's go in closer and I'll show you. Here's Naga City. Here's Mount Isurog. And this feature behind it is the Caramellan Peninsula. And I'm going to talk a lot about that tonight. And what I want to emphasize, two things here. Here's the Caramellan Peninsula, which is connected to this Bicol Peninsula. The Caramellan, that sort of canoe handle shaped to Caramellan, sticks out into, here's that 120 meter bathymetric contour underneath these islands of today. Luzon Island, Catanduanes Island, and Pelili Island were all part of this big land positive peninsula. And this part, Caramellan, you can probably see this different color in water back here that's supposed to indicate the presence of deep water on that edge. These areas, these little islands right here, shouldn't have endemic species on them. Those little islands we're part of this microcontinent, basically, this whole land bridge, this whole extension of the Luzon shelf here. And so those islands where we ended up finding species that only occur on those islands and not the adjacent mainland of Luzon were a real surprise. And the other thing I want to point out is the string of volcanoes that goes all the way down the Bicol Peninsula. 1, 2, 3, 4, 5 are visible in this picture. And the Bicol Peninsula is this fascinating place of just a row of volcanoes. And it's an amazing place for lots of other reasons, but it's a very dramatic scenery and a really cool place to do field work. So you fly in here, and the flight is a little rough when you land, and the pilot has to circle right around this mountain, Mount Mayon, and you land there and step off the plane, and it's like, the smoking volcano in the background. It's really dramatic and has a really strong impact, and you get there. And at night, it's like these beautiful, iconic Bicol land and seascapes, and you sit outside and eat some really good Bicol cuisine and have this view of the scenery, which is really fantastic. And I think you can tell from the postures of some of these KU students that they're pretty excited to pull over on the side of the highway and look down that chain of volcanoes and see that view of just volcano after volcano after volcano as you go down the Bicol Peninsula. Bicol is really cool. It has these old, beautiful Spanish churches, but it also has its own endemic culture and flavor. And one of the things that's a common theme is that people live in the shadows of these big volcanoes, as indicated by this Spanish church that was all but buried by a lava flow that came down Mount Mayon right here. And all you can see today is the bell tower sticking out of the top of the ground. And the rest of the church just got completely buried by the lava flow. And so these people live very close in proximity to these volcanoes. It's a great place that has all these specialty foods, a lot of which are around the production of this nut, pili nut, which actually forms part of the diet of Varanus olivaceus, this Bicol endemic frugivorous monitor lizard. And they make all sorts of desserts with them. And it's also a place where people like really hot food. And so if you like hot food, they have hot pepper ice cream in Bicol in the Bicol region, and it's awesome. You can get it in flavor in strengths of 1, 2, or 3 hot peppers. And I just love this poster because, again, more fun in the Philippines. Well, it's really fun in Bicol because you could go to Hawaii and go surfing, or you could just come to Bicol, or you could go climb mountains in Mount Fuji in Japan, or you just come to Bicol, or you could go, to a rodeo in Texas, but you could also just come to Bicol, right? So I just think these posters and this theme of more fun in the Philippines are absolutely true, and it's a great place to do field work. So here's the Bicol in that amazing landscape. One of the big trends in funding in this priority of biodiversity science in the Philippines is supported by the Philippine government are big research projects that focus on ridge to reef biodiversity distribution, studies from ridges all the way down to the reefs, the entire ecosystem, and the different habitat types and communities of people and species that are found along these elevational transects. And so from ridge to reef, we wanted to conduct to set up this long-term transect study with our collaborators from Ateneo de Naga University, stemming on a permanent transect that would go from the top of Mount Isarog at about 2,000 meters all the way down to the marine habitats of the Caramellan Peninsula. So the idea was to set this up in multiple different stages. There would be multiple different climbs of biodiversity surveys of amphibians, in my case, amphibians and reptiles, but in their case, other subjects as well. Multiple different approaches to Mount Isarog, spanning the saddle between Mount Isarog and Caramellan, which is a low elevation and highly disturbed agricultural matrix, but could tell us something about disturbance and land use patterns. A long, slow, gradually decreasing part of the transect that would go down the Caramellan Peninsula and slowly decrease in elevation as we cross multiple peaks in the Caramellan, and then eventual extension out into the Caramellan Peninsula, which is a heavily forested, very well-preserved national park. And then perpendicular sub-transects that would go out from these ridge lines down into the reef and into the marine areas where my collaborators are from. They're one of them, Michael Flores, is from Caramellan, and he's actually a seagrass expert and studies seagrass bed ecologies in those areas. And so he has a long history, his family's from Caramellan, and a long history of studying reef and marine biology there. And so the real interest of putting all this together into this series of 15 to 20 sites with places where we could do surveys and resurveys, because of course there's seasonal variation as well. And in the dry season, one catches a lot of reptiles, and in the rainy season, you catch a lot of amphibians. And so if you want to know something about the total biodiversity, it's important to do a survey and a resurvey, and at least those 2 different times of the year, spanning all that seasonal variation and all that habitat types across the entire Caramellan. And so this work was conceived of as this big multidisciplinary collaboration with Michael Cloris and Michael Cuesta at Ateneo de Naga. And they have real strengths in their university in social sciences and livelihood and welfare studies. And the idea was not just to focus on biodiversity, and in my case, amphibians and reptiles, and other collaborators who might want to join who have other taxonomic expertise, but also natural resource management and use, community perceptions of biodiversity, historical land use patterns, all focusing on variation and the degree of level of forest disturbance and what that's done to biodiversity across this complex structured system. Because the communities of people who live, for example, on these small islands, who are fisher people for the most part, are going to be their concerns in environmental resource use and their concerns about natural resources are going to be very different than the people who live on the tops of these mountains in these forested villages at high elevation. And so that the human perception of biodiversity was definitely meant to be part of this whole thing. Well, it turns out, there actually is this long herpetological history out there, this history of Walter Offenberg, the scientist who studied the long-term biology of Gray's monitor and wrote that wonderful book about Varanus olivaceus, at that time the world's only frugivorous monitor. He spent a lot of time out in the Caramellan Peninsula, right in that same town. And they wrote these wonderful monographs on, in this case, reproductive patterns of skinks. And his 2 sons, well, his whole family, but his 2 sons came and did some of this work with him. And I think it's Troy and Kurt Offenberg, and I've actually met Kurt years ago in the Philippines, but Troy wrote with his father this paper on reproductive patterns in Philippine skinks. And across the Caramellan Peninsula, they did things like they quantified environmental and documented elevational variation and atmospheric conditions, including the monsoon seasons. They mapped the presence and the physical position of all these different species and showed where they're most common across this elevational gradient. They did things on fine species that were ecologically similar. They showed very detailed studies on fine scale resource partitioning of those species. And they even drilled down into all the courtship biology and reproductive seasons of all these species, focusing on all these variables across several different species, and showed that reproduction was really triggered by the dual monsoon season and the 2 rainy periods that occur in the Caramellan Peninsula, which is unique because they have 2 rainy seasons there. So it's just really amazing biology, great data, all structured right in the same area. And I was really excited to follow up on some of that and do some of that work in collaboration with new collaborators and students. So here's the view of Mount Isrog from Naga City. How do you get to Caramellan? Well, 10, 12 years ago, when we tried to go from Presentacion to Caramellan, we got turned back right here and we couldn't go out to this area and see this forest, because the only way to get there then was there was no road, and you had to get a boat from Tiwi and go the long way around, and it was slow and expensive and very difficult for us with a big group of people and all of our gear. So we were turned away and didn't get to do the work we wanted to do there the first couple times we tried to go, but now there's a road. So what would be well, we'll come back to that, but there's a nice road that goes right out of the Caramellan, and there's Catanduanes Island out in the background. Beautiful, goes right through all this wonderful habitat, like high elevation montane forests in Lagonoi, and mid elevation, different types of vegetation in the forests of Presentacion, and the low elevation forests of Caramellan proper when you get out to the national park. And inside those forests are these wonderfully different variable habitats, like interior lowland closed forest, closed canopy forest, and these pandana stands that have different species of lizards in them. Beach coastal forests, which is some of the places we found some of these really cool new species of Eutropis that Anthony's been publishing on. Amazing habitats that I've never gotten to work in all my time in the Philippines. Limestone forests with intact forest on top, and a whole different suite of species that one finds in all those different environments. And then you get out to this formerly really isolated place, and there's this wonderful amenities there and a great place to stay. And you're asking yourself, how did people, where did they come up? How did these resorts get built if people couldn't even get there? Well, the answer, does anyone know? Okay, you're all dying to know at this point, right? The answer, Survivor Philippines. That's the whole reason for the building of the road. To film Survivor in this show that's wildly popular in the Philippines for 4 or 5 years now, they had to build a road in case of emergencies to get people out, in case they had to evacuate anybody. And so this resort is actually the place where they put the people who got kicked off the show, the weakest links who get ejected from the show. This is where they stayed. So the road and some of those amenities are related to Survivor Philippines, and in particular Survivor Caramellan. But right behind these beautiful resorts, which were built for that reason, is this pristine habitat that we could access really, really well. There's forest-covered karsts of Caramellan are some of those dramatic landscapes. Oh, here comes Poppy. Okay, girl. Sorry, my dog wants to say hi. Are some of the most okay, down, are some of the most dramatic landscapes on Luzon that I've ever been able to work in. The limestone is really wonderful. And as soon as you take a look at it, you know it's going to be full of geckos. And there's these wonderful, huge caves that are just like these amazing, giant ones that groups of kids could take us to with no problem. These big karst cathedrals with skylights and wonderful formations that indicates that they're big, healthy ecosystems inside these caves. And emerging on the other side, we come out to Caramellan coastal habitats, which are these beautiful habitats that show sites, again, things that I haven't seen in the Philippines because I haven't got to work in coastal areas that are not developed. And Ateneo de Naga owns a boat and can take you out to the small islands where we want to do surveys in a type of habitat that I've never been able to work in before. So motoring out to these islands is like one of those scenes in like those resort places in Thailand with these beautiful karst and just wonderful scenery. As you get out there, you go out and you see habitats that I just want to climb all over, karst and sand habitats and freshwater lagoons on the insides. So there is fresh water. You can see where they filmed Survivor Philippines right here on the shore. Rocky beach forests, and these isolated coastal habitats and fresh water right next door. And we think that the rugged, really arid karst serves as kind of a barrier and may isolate some of these habitats. But in the areas here where we've been able to do these full surveys, we find species that we certainly didn't expect to find on Luzon, and species that are either along the coast or shared with the nearby small islands. Here's that freshwater lagoon that supports all sorts of amphibian species right there. And these islands of little beach forest with endemic undocumented diversity. And just this simple thing of natural vegetation spilling over onto the beach is just not something you see very often in the Philippines, because the coastal regions are so developed. But these beaches are pristine and have natural vegetation, and they're full of lizards. Okay, and so here's a species that I don't see very often in the Philippines, but we find lots of them there. This is Daisy agrizia, one of these species that is a coastal area specialist. And I'll take you through some of the diversity and show you some of the other diversity there. At this site, we see multiple species of Brachymeles, including a fully pentadactyl one and a leg-reduced one that's almost legless. We see Tropidophoros, these crocodile skinks, which people call Burobuaya, which just means like baby crocodile. They're pretty neat. An undescribed species of Lepidodactylus that we found in the first week of being out there. An undescribed new species of pseudogecko that this description just came out and is found on these same environments as well. Anglehead lizards that turn colors when you pick them up and mess with them enough to try to take their picture. This is one of the few places I think there may be multiple species of Gonocephalus at the same spot. Pretty neat. The newly described Uranus is common in that area. Hydrosaurus, these amazing sail fin lizards, are all over the place. And your requirement, if you give a talk at Berkeley, your Draco lizard that you see, Dracospilopterus, the widespread Luzon species. And amphibian diversity is surprisingly high in this environment. Here's Sanguirana luzonensis. Here's Pulcherana similis, two species of Rhacophorus, Bimaculatus, and Pardalis, several species of Platymantis. I'll just show you two, Corrugatus and luzonensis, and then the fanged frogs that Jim and I are working on still to this day, Limnonectes woodworthy and Macrocephalus, and yes, they do have fangs. Two species of paddy frogs, both species there, and four species of Kalula, and I'll just show you Picta and the tree hole frog that's endemic to Catanduanes, the small island, and this coast, Kalulu Kukache. Snake diversity is really high here. Here's the endemic species, Boiga angulata. These beautiful yellow Ahechila there that are just gorgeous animals. Lots of Dendrolaphus of two species, or maybe three. And then really rare snakes like kukri snakes, Oligodon and Chorus, that I've only caught 10 of these probably in all my time of work in the country, and they're just beautiful snakes. Another endemic species, Boiga philippina, was there. And then this group of snakes, Oxyrhabdium leperinum and Hologarum philippinum, are now recognized as these are the two of the 5 genera that are in this endemic Philippine snake family. And this is one that I've only collected like 4 or 5 of these in all my time. And they're right there in Caramellan, Olagerum philippinum. Lots of pythons, the Philippine false coral snake, which is sister to, get this, king cobras. How weird is that? This is a beautiful little burrowing thing that looks like a coral snake, and it's not a coral snake. It's the sister group to king cobras. Pretty amazing. And then pit vipers, Drymeroceras flavomaculatus, and temple vipers are all over the place. And if you go out at night and climb around on the rocks, you see sea snakes everywhere. So just amazing diversity, pretty exciting, and leads to hopefully long ability to conduct multiple repeat study systems out here across this transect if we can go back year after year. And then the pandemic hit. We were getting some pretty interesting data, two or three different sites with respectable species counts at each places and the need to go back in different seasons. But we were finding that endemism appears to be positively related to elevation, and species diversity appears to be inversely related to elevation with a bit of a mid-elevation bulge. And in this area, we find these small islands off the coast of the Caramellan down here turned up all these new species that I wouldn't have believed it. It's not the place to go find new species. And that's, again, well, it's probably because of the Pike paradigm convincing all of us that Catanduanes and Polilio and all these little islands in between were land bridged to Luzon, and so they should have a nested subset of the species on Luzon. And why would some phylogenetically distinctive species survive on one of these tiny little islands and not even be related to the things on the adjacent mainland of Luzon? And that sort of dominant paradigm has really sort of cast an expectation over biologists in the Philippines, myself included. And so a lot of these small islands have just been skipped over. A lot of them are heavily impacted and are now coconut plantations, but some of these places and other islands throughout the archipelago, we're starting to find this pattern that I'm about to show you over and over again, which is endemic species that come out in phylogenies where we wouldn't expect them. So here's sort of a schematic of the different expectations with using hypotheses, how you might test predictions for small islands, thinking about 2 different Pleistocene aggregate island complex, a pike here, a deep water island that was not joined to either of those, and then these land bridge islands that were connected by channels beneath them that are less than 120 meters, and so they should be connected. Now, if you find a species, phylogenetically distinct species on island C, you expect it to be related to the big landmass nearby to which it's related. And this is the outcome that we keep turning up, which is the species on the land bridge island is actually phylogenetically related to species 6, 7, and 8 over here on another pike and not to the adjacent mainland. And that just defied all those expectations. And so setting up this hypothesis testing framework and asking this question just in the Caramellan, we came up with 5 or 6 answers of this result, of this species on a land bridge island coming out related to other parts of the archipelago, not to Luzon species at all. So here's an example of Draco, which does follow the rule. Here's an example of Draco in an island from northern Luzon, Draco on Batan Island, and it's nested right within, correct me if I'm wrong, Jim, nested right within these Luzon clades, the light blue clade, the dark blue clade, and the yellow clade, just right around there. And that's a case where we're just what you'd expect. And these potential distinct lineages down here in the southern Western Visayas on islands down here are nested right within this big clade of green things and are related to the things that you'd expect that are from the Western Visayan Islands to the rest of its pike. So those are cases where they do follow the rules. And here's some cases where species don't follow the rules. Here's a new species of gecko from a tiny little island north of Luzon. And you look at its position, it's related to species from the central part of the archipelago and not to the species Gecko mindorensis and Cucuchii, which are right adjacent to it on the island of Luzon. Or a species on a small island in the Babuyan Islands, a species of frog, that sister species is down here in Mindoro, and it's not related to the intervening large island of Luzon. So that's what I mean by these phylogenetically anomalous relationships in defiance of the Pike paradigm. And that setting up that hypothesis testing framework allowed us to do a lot of comparative biogeography and find species like Caramellan's own pseudogecko huncog, the species that just was described, and its closest relatives are from Negros, are from the central part of the archipelago, and from Leyte Island in the southern part of the archipelago. So this is the first photograph of the species that was taken by my collaborator, Jason Fernandez, 5 or 6 years ago, and that's a Caramellan and southern Sorsogon Luzon endemic. Here's another example of a gecko from the Caramellan. So this is the species right here. And its closest relatives are things from the northern Philippines, off Luzon to the north, or from the small islands in the central part of the archipelago. And it's not related to the species Gecko mindorensis that's distributed throughout Luzon and Mindoro and Gecko kikuchii. So we keep finding these small island, phylogenetically divergent, really distinctive species. And these are phenotypically, screaming distinctiveness at us. You pick them up, some of these things like the pseudogecko that I just showed you, as soon as I saw it, I was like, oh, new species, absolutely. There's nothing else that looks like that. And same for this gecko and lots of these other species. So they're very distinctive, and they're unrelated to the adjacent sort of mainland body mass or landmass near them. And so we think that this might reflect that these might be species that are surviving now on these small islands on the periphery of what might have been a formerly much wider distributed, wider distribution. And that maybe successive waves of colonization of other gecko species wiped out the population on the mainland, but the populations on the small island didn't get hit and have survived there today, blinking out in this last little bit of on the fringe of the periphery of their original distribution. I think that story of Wrangel Island mammoths may be very similar, right? Wrangel Island is the place where mammoths survived until 3,500 years ago on a small land bridge island off the coast of Alaska. And they survived, up until 3,000 years ago, whereas all the other mammoths went extinct in 10 or 12,000 years ago at the end of the Ice Age when people started hunting them. So their predators wiped them out, but didn't wipe out the population on the small island. Even though it was land bridge to the mainland, humans didn't get there and didn't hunt. So it may be something like that. But but now that we've started to sort of drill down on this pattern, we're looking at some other exceptions as well. So another exception to the Pike paradigm in the way that the diversity is partitioned in the archipelago might be widespread species. If you think about this, imagine a species that's widespread throughout the archipelago, a single species that's found on all the different Pleistocene aggregate island complexes. And it's a place where you wouldn't go looking for structure and for information about either trying to match distributions to the pattern or trying to test hypotheses about the predictions, testing hypotheses generated as predictions from the hypothesis testing framework of species pump mechanisms, for example. Here's Polypodates leucomystax, which is ubiquitous in the country, munching on a house gecko. And they're voracious predators as well. And in this paper that we published years ago, it sort of set the tone for me. We had this big collaboration with our collaborators in Indonesia and lots of different people who had sampling from all these different areas. And we had amazing sampling in the Philippines. And after working throughout the archipelago, I got pretty excited about, I can't wait to sequence these things. We had like 170 samples from throughout, like every island in the Philippines. And after that amount of field work, we had them from everywhere. And I was just so excited to see what relationships we got. And it was kind of disappointing. Here's all this structure and all those non-Philippine populations, but in comparison to them, the Philippines was absolutely genetically identical. And it was kind of disheartening, I thought that, I thought I would find some phylogeographic structure. And, this is the single locus, single locus phylogeography era. So maybe it's not surprising that we didn't find much variation. But across all the Philippines, it seemed like we found no structure. And so we came up with an interpretation and a story and just discussed the possibility of human-mediated range expansion. And basically asked the question of whether, if there was originally structure across the archipelago in this species, maybe it was sort of obliterated by the Industrial Revolution and agriculture throughout the country and the expansion of the coconut palm plantation industry and rice cultivation, and humans transporting these animals around all the time, which we see today. So that's where we left it. And I was sort of thinking that a lot of these widespread species, I had been collecting them as we were doing field work throughout the archipelago, thinking about someday doing comparative work with widespread species. And I sort of expected that result. And so one of those groups that I was thinking about eventually wanting to work on was Ossidozyga, these puddle frogs. And when I got that result, we got those results with Polypodates, I was like, why bother? We're not going to find hidden diversity in these things that are just the same species and that live in rice fields and are found in every roadside ditch. And this is a species that is ubiquitous in the country, and you find it every body of water. I hear them in the open sewers in Manila, right? They're literally everywhere. And so why would you expect them to be adapted to different areas or be partitioned when they would be transported around just like Polypodates? So I went to it with that preconception, and boy, was I wrong. I thought they were all the same until I caught this frog. And so here's a site where where this widespread single species, Ocedozyga laevis, throughout the Philippines, I found a spot where I found these phenotypically distinctive different forms. This is one that we found in the same valley, and this frog we found clinging to cracks in rocks and waterfalls. And they're down at the river and the puddles in the forest below, you get the normal species, Ocedozyga laevis, but up on the cliffs, you catch this thing, which is bigger in some cases and has drastically this wildly tuberculate skin, and keratinized asperities on the tubercles, and the taxonomic characters like the shape and the numbers of pads on the hands and feet and the shape of the toe discs and stuff are all really solid traditional character differences. But these are things that we collected together just 50 meters away from each other, that seem to be in distinct microhabitat differences, with a specialist that was living up on the cliffs and had these big thick toe pads that look really different than the common species that lives down in the river right below. But you could see the potential for the possibility of some gene flow between them because they're so close together. One could imagine that in a flood or something they could get mixed up, and we could potentially get admixture and gene flow between these. So we did a little survey, and for about 400 samples of Ocedozyga from across the archipelago, we sequenced 16S from across all these samples, from about 400 samples across the Philippines, and then there's some outgroup sampling just to root this network. But we found great structure, unlike the Polypodades case in Palawan, Northern Luzon, Mindanao. These are all sort of the Pikes, the West Visayas, Samar and Leyte, Southern Bicol, Northern Bicol, or even finer partitioning of the 5 major Pleistocene aggregate island complexes. And they all seem to be about 10% divergent from each other, which is getting up there in the realm of, the P distance, so the genetic distance that people use as a rule of thumb to start looking for characters to see whether they might possibly be distinct species. But we didn't want to do that because it's been criticized for all, on all these different grounds over the years. So we decided to unleash the awesome power of FrogCap, which is this modular new probe set for genomic data designed by our former student, Carl Hutter, who's now a postdoc at LSU. Carl designed this modular probe set that could be adapted for different groups of amphibians and tested it for different families. And we used the dichroglossity subset of these probes to sequence more than 6,000 loci from across the genome. Here's the details of the data. And the red dots indicate the samples that we took out for genomic sequencing. So a sequence across the archipelago and focusing on some exemplars from each of these divergent clades. So these are kind of a lot of results here together, but I'm going to put them together so we can discuss them side by sides. Here's plots of selected pairwise distances of those 16SP distances. And the ones that interested us were these 10 clades that had these distances between clades of about, upwards of 8 or 10%. But when we just sequenced a maximum likelihood phylogeny of just the 16S and did an initial MPTP species delimitation procedure, the answer comes back to strong support for potentially as many as 20 unconfirmed candidate species across this group. And so that was, again, not a result I was expecting, but we really espoused this 2-stage discovery stage and then validation stage, 2-step process of species delimitation. So we wanted to really take all the tools that we had here. And here's the results of k-means clustering analysis. And this is our astral species tree, summary tree phylogeny across just the Philippine populations. And these validation step analyses suggest, using pseudo-F and the Bayesian information criterion, suggest somewhere of about 8 or 10 species, with a little bit of disagreement as to whether some of those samples might be distinctive or not. And I'll show you this sample right here that shows that one difference between. That's the really tuberculate frog that I just showed you the picture of, that really sort of started this whole study. Here's maps of the ancestry coefficient bar plots. So these are the astral tree and the structure-like analysis with as many as up to 20 potential unconfirmed candidate species. And you're looking at the ancestry coefficients for K of 4, 5, and 6. And then those colors correspond to these positions on the map. And our cross-entropy plot suggested somewhere between 4 and 6, maybe more, ancestral populations, and then a lot of admixture between them, right? So the possibility for gene flow between some of these things. And even if we go with, I should mention, these are the SNPs that were mined from the FrogCap loci exons. introns. And so even if we go with the most conservative 4 potential species here, you can see lots of room for admixture. And if you go up to the more liberal 6, these some of these get called some of these clades that get called as distinctive genomic sort of centers of variation or ancestral populations do correspond perfectly to the biogeography and the Pike paradigm distribution pattern expectations. I hope you can see the color, the shading here. The purple shaded island is the home, of one species. The central West Visayan Islands in blue, Palawan in green, Mindoro Pike in red, and then Luzon in brown here. And pretty good correspondence of these potential species as diagnosed with genomic data, correspondence to those distribution expectations of the Pike paradigm. Here, these are the ones that sort of sealed the deal for me here. This is a PCA analysis of the SNP data, and we replace the dots with the pie charts of the ancestry coefficients from that S and MF analysis at K4. And you can see 4 distinct clusters, and then this network cluster from NetView defines, again, 4 distinct clusters with the possibility of these intermediates in between. Those are pretty exciting to me because I look at these things like a population here in Western Mindanao that seems to have almost fixed proportions of admixture across multiple different parental type species. I see that and I start to think about hybrid speciation. And so one of the things we're going to do to follow up on this is go back to these heavily admixed populations and get these kinds of data for another 15 or 20 individuals from those populations that may allow us to address the possibility of these potentially being related to hybrid speciation. And I'd love to talk to people more about that if we have time at the end or after my talk. But in any case, the intermediates between these 4 or 5 clusters here track to the hybrid, the individuals that are identified as hybrids between the parentals. So here's B right here from Western Mindanao as a potential hybrid between the cluster of A and C, 1 and 2. And so the hybrid index maps out exactly the admixed individuals that we hypothesize might be the individuals that are combinations of different ancestry that involve different parental genomes. And the parental, the ones shown here in NetView are these parental reference populations that are the 4 clades that seem to be entirely pure. And those ones are consistent all across the K-means, PCA, NetView, and SNMF analyses. And then the clincher here was this species network tree that was this inference from 3,000 intron derived gene trees and identifies 7 reticulation events that track back exactly to the intermediates between these parental species over here. So it looks like a clade that had the requisite 4 or 5 Pike endemic species, and one that has now had lots and lots of admixture, but we can still see the signature of these pure parental species. Pretty amazing. Last thing we went back to, I'll show you, is we went back to the question of these P distances, and we looked at the relationships between the mitochondrial 1 gene relationship and a genomic distance metric from across all those genomic data that I just showed for the non-admixed clades versus the admixed clades where it looks like there may have been gene flow. And I think you can see the difference here, a much stronger relationship for the parental clades in between the 16S mitochondrial distance and the full genomic genetic distance across these individuals. And this just suggests one more time that people ought to just be really careful about interpreting those big long branches in phylogeographic studies and those singletons that come out on the end of long branches, and people jump to the conclusion of, whoop, it must be a cryptic species. And this would be another case, another reason why you just really would want to interpret those with caution if it looks like there might be the potential for admixture and gene flow between species that you're focusing on. So I'd like to wrap up here and end by talking just a little bit about the criticisms of the Pike paradigm, and they're all the things that you could come up with, right? It's unrealistically simple. Common mechanisms are just kind of hand-wavy. It doesn't incorporate things like demographic differences among the taxa. It's a pure vicariant story for the species pump mechanism within the pikes and doesn't really get at some other aspects of biology that we'd want to get to, et cetera, et cetera, et cetera. Basically, does it boil down to just a straw man hypothesis? And these things are all true. They really are, right? But here's another hypothesis testing framework that that served its purpose for many years. The Hardy-Weinberg Equilibrium. When was the last time that you found a population that was in perfect Hardy-Weinberg equilibrium? And I mean, I used to analyze these kinds of data in Sheldon Gutman's lab, and I found some that were pretty close, but most of the time it's when something deviates from the equilibrium or the hypothesis testing general sort of neutral predictions that we have something interesting to say. That's also true of the equilibrium model in biogeography, which is near and dear to our hearts, right? And it was after this, I mean, we don't really expect all these islands to be in perfect dynamic equilibrium based on just these factors. But it was after the formulation of this model that everyone went out to test the predictions of the equilibrium model of biogeography, that we had this wonderful sort of renaissance of all sorts of new trends in biogeography that we learned when things deviated from the model, right? So in conclusions here, I guess to wrap up, I'd just like to, today I've shown you some coincident patterns and species distributions that defy, but also teach us these simple things and interesting things about what I consider to be a simple, elegant hypothesis testing framework that served its purpose for a lot of years. It's also, this is a really important part, it's an entry-level starting place for a lot of students. And I still go to this example with graduate students when we're trying to design projects. And it's a great way that the public and lay public and newcomers to the archipelago and to the Philippines can get involved and think about looking for exceptions to the rule. And looking for patterns and trends that allow them to discuss generalities about the evolution of the organisms. It's those exceptions to the rule that actually remind us of the existence of the rule, right? And so revisiting those studies with new and more powerful data is just downright fun, and ultimately teaches us that biogeography is more fun in the Philippines, and that's definitely really true. So I want to thank my collaborators, Ateneo de Naga, Michael Flores, and Michael Cuesta, who really welcomed me to be called to do this collaboration with them, and learning new ways to do multidisciplinary collaborations has been really exciting for me. It's something that's taken me out of my comfort zone and has been really, has been an exciting period. Their funding combined with our funding has supported all this. And we've gotten a lot of support from both institutions for this institution to institution long-term collaboration. I'm grateful to a large group of people here in Kansas who've given me feedback on the system and have gotten interested in different parts of the system over the years. There's a big group of people who like to talk about these things here in Kansas. And then there's this great enthusiasm that I get from the government in the Philippines who support my request for permits and help me with my overall relationship with the government and with the National Museum of the Philippines. But in particular, it's these small offices in the provinces where I really see people who are just really dedicated to what they do and who inspect our specimens and help us with our transport permits and make sure that we cross every T and dot every I, because that's so important. And I also really appreciate their enthusiasm for the exciting research and the new discoveries and things that we've been able to find along the way. I love this picture because, the last thing I say is this field crew that have done all this field work with us for over the years. I like this picture in particular because it's like my dream team all in one place. There's Enteng Inhente, who is the person who took Jim to catch Draco on his first trip to the Philippines back in 1996, maybe, long time ago. Arvin Diezmos, who is now who's my long-term collaborator and who I met in 1990 when I first went to the Philippines, and he's now the curator of herpetology at the National Museum of the Philippines and a preeminent herpetologist in the country. Mary Tess Sangila, who's with us tonight, who has been my collaborator ever since her spending some time at KU as a visiting scholar, and then now as part of all the research that we do in the southern parts of the archipelago, and who's a professor at Urios University in Butuan City. He's been a big part of all this work. And then this guy over here, Jason Fernandez, I first met Jason when he was 5 years old, when he came up with his father, who was my field assistant at the time, and stayed with us for a night on Mount Isarog. And this was the first night that Jason, little 5-year-old guy, got to spend the night in the forest in a tent. And he credits that memory of that time that he came up to spend the night with us in the field as to one of the reasons that he really wants to have this job where he gets to go do those things and catch animals and live in tents and spend a lot of time in the forest. And so I like this picture because we clown around a lot, and here we are trying to act pretty tough. But he's a dear friend who's been involved in all this research. He's the comic genius behind our field crew, and he's also a great photographer. And he's the glue that holds together this amazing crew of Bicolanos who do all the collecting and help us with all the field work and set up the transects. And none of this would be possible without them. So thanks to Jason Fernandez. And ultimately, we're just trying to emulate this example of this collaboration between Walter Brown and Angel Alcala, who for ever since the 50s until the late 90s, just were partners in all of their research. Here's a quote that Walter Brown said to me in 1998, right before he passed away. These two did everything together, and they published more than 75 papers together. Angel Alcala is one of the premier and most eminent scientists in the country right now, and all throughout their career and some politically divisive times, they stayed together as close collaborators, and they really set the example of international collaboration, which I think really transcends divisive politics, and these two also just really transcended the racial and cultural stereotypes that sometimes characterize the sort of love-hate relationship in politics between the United States and the Philippines. And so they're transcending that, and rising above that has been a big inspiration for us, and has been something that we've tried to emulate and take as sort of a whole, sort of a whole atmosphere of our collaboration. And let's face it, if they could get through the Reagans and the Marcos era, then we can get through Trump and Duterte. So I'll stop there. Thanks very much again. I really enjoy the opportunity to see all of you, and I'll be happy to take any questions.
Should we unmute and thank Rafe?
Unmute.
You're very good.
Rafe, may I start with a general issue?
Please.
A lot of your talk, you kept focusing on how and you ended up with the exception reaffirms the rule.
Well, in a sense.
I'd like to point out that the only thing an exception does for the rule is disprove it. Because the definition of a rule is it's a rule. And if there's an exception, your rule was wrong or incomplete. And this makes sense because historically, the term prove meant test. The real expression is the exception tests the rule. It never proves it. It can only disprove it. So
yeah, I totally agree with you.
I think you're trying to twist too much to fit a term that is misunderstood. It's one of my favorite examples of how language can sort of lead us astray. And if you ask dozens of your colleagues, what does it mean? I assure you virtually everyone will try to explain how the exception proves the rule when in fact it doesn't.
I absolutely agree. I don't think we can approve anything here. And just that phrase is one that, yeah, I agree with you, gets twisted around.
Well, the phrase is correct if you take it for what it means to test the rule.
Right.
So can I just
aside from that, it was beautiful, Dan. I didn't mean to dispel.
Thanks, Paul.
So I think that we should maybe talk about what the procedure should be, because there'll probably be other questions. And so why don't we have a hand raising option here for the questions that follow Paul's. I can keep track of that. Questions for Rafe?
There are a bunch in the chat.
There were. I'd love to let people ask them. Otherwise, I'm going to ask a question.
More fun in the Philippines.
I'm trying to figure out how to raise my hand, which I don't know why I can't find it.
You have to show the participants, right? Or do you have to show the chat?
Show participants.
Because you're a co-host, you can't raise your hand if you're a host usually.
I'm not allowed to raise my hand. I guess I just have to talk over people.
Go for it, Becca.
Was there someone else or can I go?
You can go.
All right. I have two questions. One is about the site you showed that was developed for Survivor. Are there like parks there? Is there some effort to preserve the beautiful, pristine forests?
Absolutely, yeah. It's a and that's part of, I think, the motivation of Michael Cuesta and his family, and in particular the barangay captain who who presides and has been such a proponent of Caramellan biodiversity. There's a lot of concern about all the people, all the tourists. It's a big adventure tourism site. And it's right on the national park. And so that national park now has been formalized. And so that is very well protected. And that's the big area, the big forest area at the tip of the peninsula. But still, there's just a lot of concern about the sustainability of all those people, all that traffic, and all the food, and all the impact on the fisheries. And so these conversations are very real. There's basically some structure being put into place in terms of new ordinances in the municipality that control the number of tourist boats that can travel there, even, and try to bring things back to a sustainable level. So there is decent protection, and there's great people who are very concerned about that, and that's a big part of the motivation for the long-term transect study, for sure.
That's good news. My other question was related to your SNP analyses. I was curious because I've seen some like mixed ancestry signals too on these long branches. First of all, was your 3000 SNP data set, is that with 0 missing data?
I have to go back to some of the details here. That one, yeah, actually off the top of my head, I don't know the answer to that question. That may be why we're down to 3000 SNPs.
Yeah, that's what I assumed, but I know like my data I can't analyze without missing data.
Mhm.
And I also have really weird signals of admixture that
They're
it sounds
associated.
like yours make sense, or you've made sense of them. So I was just curious, kind of.
Well, it's tempting. I mean, some of those distinctive divergent branches were the ones that were phenotypically distinct, and so, I was ready to believe in those being distinct species, and that signature of admixture came out as a real surprise. But then when I started seeing it in what seems like not fixed but equal proportions in other individuals in those same populations, roughly all with the same proportions of parentals in them, there's this hybrid speciation theory that suggests that two of the ways in which hybrid speciation can happen is sort of instantaneous production of a third species from two parentals. One can be sort of mechanistic at the genetic architecture level, or chromosomal rearrangements that makes the hybrid instantaneously non-reproductively compatible with with the parentals. And then the other is the possibility of external factors, things external to the organisms that might basically make that the most adapted phenotype. And so like isolation on a small island, and this is something that's been really well studied in plants and not so much in vertebrates. So that's one of the reasons why we're going back to that question with lots more data of those individual populations. But it just got me into literature that I haven't spent much time. So I'm anxious to hear people's ideas about it, or if that makes any sense. But some combination of parental lineages that turns out to be the combination of parentals. If we do find that across the population on some of these particularly small islands, then I would start to believe in the suggestion that hybrid speciation can happen as a result of external factors.
Yeah, I guess I'm just curious to be careful about missing data and allelic dropout, especially on those long branches, because you could easily picture how certain loci are more likely to be retained.
Right. Yes.
But yeah, I'm curious. I'll be curious to see where it goes.
Okay, thanks.
Rafe, I'm just curious if I can follow up on the same one, on the same topic. I'm curious if was all of that admixture results, were those all from this this astral sort of post-astral analysis, or have you actually used like a straight up structure analysis to look at that?
Let's see.
Because that was an unusual, like I haven't used that function of astral to use these gene trees to make an admixture assessment after the fact.
I'm wondering if we did in earlier preliminary analyses, and I don't know the answer to that. I should definitely.
I'd be curious to know if you get the same result with that sort of mechanism. And the other thing that I would ask is given that you have these loci, these sequence loci, it's a data set that's ripe for using like a GFOX type approach to directly measure whether or not there's, admixture indicated between some of these lineages where you have these shared alleles turning up, and to see it as another way of evaluating that part of your hypothesis.
Excellent. Thanks.
Okay, can I ask my real question? I
How
want
much
to ask
fun
a
is it?
question about the biogeography, actually.
Sure.
So about the Caramellan Peninsula and its relationship to the Bicol Peninsula, because most of the people, the people who have done Philippine biogeography that are in the audience will know this, but a lot of people won't really know just how distinctive that Bicol Peninsula is, and the fact that it was a separate island,
Right.
separated from Luzon, and maybe not even close to Luzon during an earlier part of its history. And so there's obviously a lot of endemism on the Bicol. And so I'm curious if these small island endemics that are found there would have been expected on the Bicol Peninsula if you haven't found them there yet, but because most of mainland Philippines have been kind of wrecked, that maybe you can't find those things, but where you still have some decent habitat in these, in this coastal zone, for example, that the species still persists. If you think that might explain any of you don't find them on, it's not the same species you'd find on Luzon, but maybe it's the same species you'd find on Bicol, which isn't necessarily expected to be close to the adjacent Luzon.
Yeah, I get your point with reference to proximity to the West Visayan Pike, the central Philippine islands. But maybe I didn't say this. The pattern of these small island endemics were sort of up until about 10 or 15 years ago. That we knew of some, like Gigante Island in the central Philippines, right off the coast of Panay, has an endemic species of gecko and endemic platymantis on it, and they don't occur anywhere else. That's right off the coast of Panay, and that's not where you go to look for a really distinctive, phenotype in a gecko and a frog. And what is it about that island? It seems that it's the limestone habitat that's allowed them to stay and survive the full removal of the forest off that island. So there are these other cases of known species. And when we plugged those into phylogenies, they came out in places that didn't match, that were not related to the central Visayan pike. So I guess the part that I maybe didn't say clearly was that this pattern is turning up all over the Philippines. And it's true of small islands in the north.
I got that.
Okay.
But I was asking specifically about these Caramellan Island things just because of their proximity to Bicol. And
it could be, yeah.
Have you had a chance to sample the Bicol thoroughly at this stage after all these years?
We're getting there. I mean, that's part of the reason why I'm really interested in the rest of the transect. Herpetologically, Mount Isarog, which is a very famous national park and has been very well studied for mammals and birds. And there's only been one little study in 2001 and one little survey in about 2014. And the second one wasn't allowed to collect any specimens. So there's not even like a complete species list for that volcano. And so other nearby volcanoes like Mount Banahao, for example, has 13 sympatric species of platymantis on it, but there's only 4 known from Isarog, right? So it's undersampled. But because of my last sort of generations of grad students who had targets on the Bicol Peninsula, we did come up with some pretty good general sampling across the peninsula and have hit a number of those volcanoes. The other problem is that there's the human population on Bicol Peninsula is very, very high. It's very dense, and the coastal areas are all developed. And so there's these national parks that are kind of fiercely protected by the protected area management boards that have jurisdiction over those national parks. And so it's very hard to get permits to get in there and work inside those parks. And so actually, ironically, the parks have become these areas when you look at the distributional data of you download a bunch of occurrences from GBIF and iNaturalist and plot species distributions across the Bicol, you notice these big pockets where there's no data, and that's the national parks, right? So they're just really understudied at this point, and people really need to do the proper biodiversity surveys that would allow us to answer that question that you just posed for sure. And that's something that the Philippine government requested of me, that I would prioritize working inside of protected areas in those parks, but the protected area management boards don't answer to the Manila central office. And so we've had a little bit of a difficulty getting into some of those parks. But
left hand meet right hand.
Yes. But we'll get there. We're in this for the long haul.
Do we have other questions? Matthew Van Dam. Hey, Matthew.
Hey, yeah, so I had A couple quick questions. So at least looking at the insects, particularly the beetles, the weevils from the Philippines, they all seem to have colonized out of Papua New Guinea, or not Papua New Guinea, but New Guinea Island. I was wondering if there's similar sort of patterns with the amphibians and reptiles. And I guess, so that's question one. Question two is, I've worked a little bit in Mindanao, and recently there's areas where it's pretty difficult to get around. I don't know how have you been able to get around in southern Mindanao at all? Okay, thank you. It was a great talk.
Thanks. Yeah, there are some Papuan elements in the southern Philippine herpetofauna, like snakes of the genus Stegonotus and Tropidinophis. Those are sort of things that have the bulk of the diversity in Papuan regions. Frogs like the genus Oreophrine has 2 endemic species in the Philippines, and they're definitely related to those ones from Papua New Guinea and that whole eastern Indonesian concentration of diversity. So there are some Papuan elements there, but it sounds like a lot more in some insect groups. So that's really interesting to me. I'd love to hear more about it. And then the answer to how one does work in the Philippines is to have great collaborators like Tess Sanguila, who's down here. I don't know if you can see her. I don't know if you can see my cursor. I guess I'm not sharing. Who just can run this research program out of the places where it is safe to be and safe to work into nearby areas, and have friends and collaborators and colleagues and a professional network throughout the rest of the island. There definitely are some places, and when conflicts emerge where we can't go. But again, if you view it sort of for the long term and eventually want to get sampling from Western Mindanao, you eventually can if you wait for the right moment in time, which is like right after the elections when everything's safe and everything's calmed down. And you may be able to think about that years in advance and plan your permits for those sites right after the election. Those are the sort of strategies that we've taken. And also just going, mostly in groups of all Filipinos and just sort of not attracting attention to yourself and with big groups of foreigners who don't look like they're from around there. So that's the way. I do.
Guards.
What's that?
Armed guards.
Armed guards. We have done some of that, and there's that picture of Tess and that armed guard, which was, gosh, where was that? It was when we were trying to get into Pagadian, and that was a case where we had permits and we'd set up this expedition for more than a year and planned all the permits. And I flew a big group of students and collaborators and my whole field team down to Mindanao, and we got turned back in one day and had to give up all those plans because a security concern had swelled up. And so I think the key to working throughout the Philippines is to sort of always has plan B and have plan